Hello everyone, I'm Zhengkun from University of Florida. Today I will present my current research, the blueberry yield estimation through multi-view imagery with ULV-8 object detection. In the past decades, the blueberry industry in US has been growing rapidly and creates huge commercial values. In 2021, the United States blueberry industry reached an impressive $1.1 billion. Accurate estimation of blueberry is important for crop grading and improve management practice. However, there are small sites and dense clustering of fruits, along with occlusion caused by leaves and trunks, make blueberry detection difficult. There are several existing approaches to estimate blueberries, but so far most of them are limited. Many methods rely on manually sampling, which is time-consuming and labor-intensive. Other approaches are relying on remote sensing techniques, but the large-scale measurement is not accurate enough and not suitable for small plot yield estimation. To address this problem, we developed a multi-view sensing platform to coverage the blueberry plants as much as possible. By integrating multiple perspectives, we aim to overcome the challenges posed by occlusion and background similarity. Based on the data collection, we will make a dataset for supervised model training. Actually, the ULV-8, the state-of-the-art detection model, was used for the blueberry detection. With detection results, the number of predictions will be used for the yield estimation, and the location of the predictions can be used to generate the density map to show the spatial distribution of fruits. As for the yield estimation, we will compare the prediction from the single view and the multi-view regression, then validate the effectiveness of our approach. We collected the data and tested our algorithms in IFA's Plant Science Research and Education Unit. The field is planted for blueberry breeding. There are different genotypes planted here, and for each genotype, there are only 7 adjacent plants are planted. The figure shows the platform we collected the data. So it's called MassX. The robots move with the 0.2 meters per second. And as you can see, there are several cameras mounted on the robots to capture the image from the top view, the left view, and the right view. Those cameras include digital cameras, the Pi cameras, as well as the RealSense RGBD cameras. Except to the images we collected with the robots, we also include the images collected with handheld equipment from 2018 to 2023. Also, we collected several open-source annotated images from RoboFlow, an online web-based annotation tools. The blueberry annotation is extremely time-consuming. Most of the image contains over than 200 blueberries. Although we use the AI assistance annotation tools, but modified the labels are still taking about 120 hours. So there are several images here. I show what the dataset looks like. Totally, we got about 350 annotated images for model and training. Besides, we use the image augmentation techniques to extend our datasets to 12 times, including the rotation, flip, mosaic, crop, brightness, and blur. After we get the annotated dataset, then we trained the ULV-8 the state-of-the-art model for object detection on our customized datasets. And this figure shows the architecture of YOLO v8. We compiled the different versions of YOLO, including the YOLO v8 and YOLO v5, as well as the different configuration of the model, including the model size and the model image input. So overall, the YOLO v8 performs better than the YOLO v5. And as for the model size, the largest model size YOLO v8X achieved the highest performance with the 77.3% of MAP at 0.5. But it's only slightly better than the smaller one. What interesting things is the larger image input achieved higher performance, about 7 to 17% than the group with smaller size. So finally, we will use the YOLO v8X with the largest image size input. We tested the YOLO v8 models on the real-field scenarios. Specifically, we captured 36 images more from 12 plants. Those plants with the different genotypes. And then we did the prediction annotation regression. As you can see in the figure, the R² equals 0.87. And most of the images, the mean absolute percentage error is lower than 25% and focus on lower than 20%. This is an example of visualization of the detection results. The different color bounding box represents the different types of detection tags. The green, blue, red represents the true positive, the false positive, and false negative case. As we can see, the most common failed cases are focused on the overlap clusters, the leaves occlusion, and shadows or low contrast errors. Now, we have already evaluated the detector performance, and it can achieve about 80% of blueberry in the image. But what's the gap between the prediction and the real number of blueberries? We tested the detector within 12 plants. For each plant, we manually harvested and counted the number of blueberries, and then calculate the number of annotations from the corresponding 3-view images. We define a matrix, the visibility, to describe the ratio, the number of annotated boxes, and the number of manually counting. This figure shows the visibility results. So even the top view only can see the 52.7%. Also, the visibility differences among different genotype plants are pretty large. So even the gap more than 30%. Considering the limited visibility, how can we fuse the multi-view information? According to the experiment, the overlap among the 3 views are not enough for image and matching and 3D reconstruction. So in this case, we only fuse the 3 views detection results, actually the number of the predictions, with linear regression. Here, the result shows the R² is 0.772. And then we did the prediction and manual counting regression to evaluate the regression approach. So the multi-view approach achieved the highest R² value of 0.77 and the smaller mean absolute percentage error of 24.63%. This video shows our pipeline working on the real field. We deployed the YOLO v8 model as the detector on top image sequences and generate the density map based on the detection and results. Overall, the results is impressive. As for the conclusion, the YOLO V8 detector presents acceptable detection performance on blueberry detections. Occlusion and low contrast rate are the main reasons of fail cases. Another factor is image resolution. The YOLO V8 may perform bad on low-resolution images. As for the multi-view regression, it achieved the mean absolute percentage error of 24.6 and the R-square of 0.77, which is 5.2% to 50.7% higher than the single-view approaches. However, the raw linear regression cannot reflect the visibility factors, especially the occlusion level. So in the future work, we will improve the multi-view fusion approach, as well as add the size estimation functions to estimate the weight of the blueberries. This project, funded by the University of Florida, IFAS Lift AI seed grant. Thanks for your listening. That's all for my presentation.